Introduce Dr. Jerome Nye. I hope I've pronounced your name correctly, from UPF. Jerome conducted his PhD at UPC in Barcelona on finite element modelling of the lumbar spine. And then he moved on for his postdoctoral positions, first to the AO Institute at Davos, followed by the Eindhoven University of Technology, before returning to Barcelona, where he headed up the biomechanics and mechanobiology group. In 2015, he moved to UPF, where he's a tenure track professor. And today he's going to present some of his work on systems mechanobiology. So over to you, Jerome.
Okay, so thank you very much, Emma, for this kind introduction. I'm in a delicate position because I'm speaker. I'm also as a chair of the PhD summer school. So it's part of my responsibility to respect the timings. So please forgive me in advance if I decide to switch slides at some point in my talk, and we can go on with the discussion anyway. So in order to start, actually, I would like to start at a very large scale, at the population scale. I'm talking a little bit about diseases, and more specifically about non-communicable diseases. Non-communicable diseases are those diseases that are not bacterial, are not viral, and cannot pass from one person to another. In order to talk about the burden of this non-communicable disease, so we take as descriptors the disability-adjusted life years, which is the sum of years of life lost due to premature mortality, plus the years lived with a disability. So if we look at the evolution since early 90s up to nowadays, more or less, of the dailies, so according to the World Health Organization, so we can see that dailies are basically are steady, which is kind of normal because so people live longer. So actually the years lived with disability increase. But if you standardize then the dailies by age, then you see that we have decrease. So that's very good news. In general, the population, as the world population, is going better along the years. However, now, if we focus on the percentage of dailies that are due to non-communicable diseases, the figure is different. So here you have the world map in 1990 of the percentage of dailies due to non-communicable diseases. And here you have the same map, but in 2019. And you see that worldwide, actually, the percentage of NCD and the contribution of NCD into dailies has increased. And that's not very good news, because it meant that independently on whether we're talking about higher income or lower income countries, we're not controlling actually these non-communicable diseases. So if we look at the top 10 of leading causes of global dailies, and then we filter this list by NCD, meaning that in this list of 1 to 10 here and this list of 1 to 10 here, I'm only reporting non-communicable diseases. So first, you can see that from the 90s to nowadays, so we have a larger amount of non-communicable disease and in the top 10. So according to previous slide, also we see that we have non-communicable diseases that are related, that involve mechanical factors. And actually the rate of non-communicable disease that involve mechanical factors has also increased, has doubled actually, from 20% to 40% on this in this short list. So I would like to seize the opportunity to rethink a paradigm that musculoskeletal disorders are an aging problem. Musculoskeletal disorders stand for the typical group of non-communicable disease where mechanical loads are involved, obviously. If you look at the same top 10 list of dailies and filtered by non-communicable disease, by age group of population, you can see that already from 10 to 24 years of age, actually low back pain already ranks 7. And this is also the only non-communicable disease associated with mechanical loads. And if you look at the older population, yet not so old, so below 50 years of age, so people will still have a lot of years of active professional life and needs to manage many different things in their life. So you see that not only low back pain, so rise up the ranking, but also other musculoskeletal disorders appear. The typical view that musculoskeletal disorders are a natural effect of age is kind of dangerous because I think it drives away the attention on the burden that causes these disorders already on an active population that can heavily suffer from chronic pain and from disability. And so no, I'm stopped specifically talking about musculoskeletal disorders, and I'm coming back to load-bearing tissues involved in non-communicable diseases. So we're talking about rheumatology problems, so here in musculoskeletal tissues, but we're talking also about atherosclerosis that would be responsible for ischemic problems, for stroke. And we can also talk about lung tissues that are involved in emphysema, which is the most severe form of chronic obstructive pulmonary disease. So, and in all these tissues, what we have is that there are external mechanical loads acting on the organ. These loads are transferred to the tissues of the organ through the tissue infrastructure and composition. And then they are transferred to the cells, again, through the tissue infrastructure and composition. And the way cells see the load, of course, will shape the cell activity, and cell activity will trigger certain biochemical signaling. This biochemical signaling will generate autocrine cell stimulation and paracrine cell stimulation, meaning that cell will be able to stimulate itself with the biochemical factors it has secreted, and it will also stimulate its neighbors. So actually, we enter here in a feedback loop. Depending on the cell activity, so we will certainly have tissue turnover, either maintenance or degeneration of the tissue, and the way external loads and the way tissue loads are transferred will be altered. So we enter again then the loop iteratively. And there is specifically in avascular tissues like cartilaginous tissues, and more specifically, I'm thinking about the intervertebral disc, which is a large one. You have another way of transfer of mechanical load effect to the cells, which is through mass transport. The tissue is like a sponge, and when it deforms, it mechanically collapses, and the pores gets closed. So if the pores get closed, the volume of water through which important molecules can travel is reduced. So the transport of this molecule is actually reduced. So the biochemical environment that the cell sees in order to regulate its activity is modified. And it's particularly critical, for example, in case of nutrient availability in avascular tissues. And then again, so we enter basically in a loop. So cell activity, so cell consumption of these molecules will depend on the current concentration, and will modify, so cell consumption will modify so the current concentrations depending on to what it perceives, which will then again affect the mass transports depending on the mechanical loads. So here we see that we have an interaction, and I've put color code of physics or multiphysics phenomena, of biological phenomena, And then I have put specific color code for what I have called indirect mechanotransduction through these mass transport effects, and this direct mechanotransduction, which is direct sensing of mechanical loads by the cells. So actually, mechanical loads are part of a relatively complex system. They are not on their own, and their effect will depend on all the on the selectivity and surely on the gene information of the cells, but also of the biochemical environments that will have obviously external perturbator, depending on possible endocrine problems, depending on circadian cycles, depending on whether you have low-grade inflammation, etc. There are many reasons for which this biochemical signaling can so evolve on its own and then affect this whole loop. And this is where numerical models, theoretical and numerical models, become extremely relevant. So I just would like then to give a bit of overview of what were the early observations and the first models. So Ralph talked a little bit about this, but in around 1860, so an anatomist here from Mayer noticed that the architecture, the trabecular architecture within the bone, which is the internal architecture of the bone, was following a very specific pattern. At the same time, without having any relation with Georg Hermann von Meyer, there's a civil engineer, Carl Kuhlmann, that was using and popularizing the graphical determination of internal forces in mechanical structures. And here you see an example of a crane. And at a scientific congress, so von Meyer and Kuhlmann came to meet and started then to compare the internal architecture of bone with the graphical lines of internal forces that Kuhlmann was determining. And then they decided to take a crane and just modify the loading and put mechanical loads as if it were a femur, so supporting the pelvis load on the top, basically. And Kuhlmann recalculated the lines of the internal forces. And he came relatively to something that was more and more like the lines, the internal bone lines of the femur. And a little bit later, a surgeon, Julius Wolf, that Ralph mentioned, retook these results, retook the collaboration with Kuhlmann, and then redid the determination, the graphical determination of internal forces, and this time by taking the geometry of the femur. And then he could find a relatively good match. So I know if you took then modern computational methods where continuum mechanics is implemented, and you can see that if you simulate reasonable loads on a bone structure, when I'm saying reasonable loads that would be relatively like physiological loads, you're calculating principal stresses that have specific direction that almost directly then compare with preferred orientation of the trabecula, so in specific locations. Now, if you're talking about bone remodeling, as Ralph mentioned, the problem is a bit different because here you have a static representation, here you need a dynamic representation. So, and at the beginning of the 21st century, so Hesketh and colleagues proposed and developed these mechanobiological models where actually the mechanics, the calculation of strain energy density within structure was interacting with specific cellular functions. And here you recognize the osteoblast and the osteocytes that Ralph was talking about, that are responsible to sense the mechanical loads to stimulate the bone formation depending on specific thresholds. And if there are cracks or if there is no load, then another type of cell is activated and take care about bone resorption. This obviously modifies the structure, so it actually modifies mechanical loading that osteocytes and osteoclasts will feel, and then we enter into a new iterative loop of computation. So if you look at the calculations, so they first took a regular grid and they tilted the load with respect to the main orientation of the truss and started the calculation. And so we see that actually you optimize the amount of material and you globally then reorient your internal structure according to the load. Meaning that strain energy density, so according to this law or to this algorithm, is a reasonable descriptor of cell stimulator. Now, if we look at clinical reality, things are a bit different. So here we have a clinical case of stress shielding. I don't know if you know it, but this is very well known in orthopedic biomechanics, as when you implant into a bone a structure that is stiffer than the bone, the structure might take over most of the stresses, and then the bone doesn't see enough stresses, so its cells that are mechanosensitive recognize that they are becoming useless, and we start to have bone resorption. Okay, so this is basically according to this scheme, so the activation of this osteoclast. And we can, through radiological follow-up, through dual X-ray absorptiometry, so you can see, so after implantation, so where the bone has been resorbed, so these negative numbers here, and where the bone has increased. So basically, bone has been resorbed almost everywhere. And to do that, then these people did that for 3 subjects. And they made a corresponding finite element study, so computational mechanical simulations, where then they calculated the strain energy density and other descriptors were more or less realistic mechanical loads, and checked whether they were able to reproduce the clinically observed loss of bone. And we can really see that depending on the subject. So here, for example, you have subject 1, 2, 3, 1, 2, 3. You can see in this area that some criteria will work relatively well or will completely fail. So it means that you cannot really apply this kind of deterministic law of bone resorption, at least at the macro scale, to reflect the clinical reality and the variability among the patients. So actually, if you come back to the literature, already in 2000, so Haskins, who was then the author of this mechanobiological models, so was already saying that the similarity between trabecular orientation and stress trajectories is circumstantial, not causal. And there are no mathematical optimization rules for bone architecture. It's just a biological regulatory process producing a structure adapted to mechanical demands. And that responds to an evolutionary process. So actually, if we come back then to these schemes that I had introduced before, so you perceive then through these observations this kind of complexity. So the whole regulation system that encompasses biochemical signaling, mechanical signaling, etc., has properties that its different parts don't have on their own. And the properties of the whole depend on the interaction of its parts. But the interaction of the parts are not predictable submodels per se, because they can evolve according to a lot of side effects. So if I'm sticking to engineering, for example, it would not obligatory respond to the principle of thermodynamics, not the increase of entropy and the decrease of the enthalpy, etc. Meaning that we have actually here an emerging phenomena that corresponds then to the behavior of this whole system that might not be descriptible then through any law. So let's go back to the data and see what the data say. So if we look, for example, the determinants of intervertebral disc degeneration, the intervertebral disc is a very good example because it's a typical structure that bears a lot of mechanical loads within the body. And these are the results of a study that studies what would be the main leading causes of intervertebral disc degeneration in twin studies, so in homozygote subjects. And it's very interesting to see that in the relatively upper levels, so they see that familial aggregation that would correspond then to the genetic material has a very strong influence. Physical loading as a unique leading cause is relatively modest. Now, if you go down the levels and you start to look at the intervertebral discs that bear larger amounts of loads, you see that the genetic information doesn't suffice per se, and about 70% of the intervertebral disc degeneration so remain unexplained. Curiously, while the mechanical loads, the amount of mechanical loads increase, so the physical loading as a leading unique cause also increases. And the reason it's because it basically starts to interact more importantly with the genetic information, with the biochemical cell stimulation that emerge from this genetic from the baseline activity of the cells, the constitutive activity of the cells, and the mechanical loads that will depend on daily activity and on chronic loads, and on other factors, whether the person is obese or is not obese, then you will have more inflammation, more adipocine, whether you have comorbidity or not. A lot of things will start to interact with each other, and then you basically, in the case of non-explained, unexplained disorder. So we've taken osteoarthritis as an example in order to see what controlled clinical data, which information controlled clinical data could give us, and whether we can simplify these very complex interactions on a very high level just by trying to make correlations between among observable factors. So we had a cohort of 47 women with moderate knee idiopathic osteoarthritis, so matching Kelvin 2-3 at a hospital here in Barcelona, a public hospital, Hospital de Mar. And so we had 8 clinical factors, so function, pain, central sensitization, catastrophism, anxiety and depression, synovitis, and effusion. And we also acquire with some gate analysis for all the patients. We used two-vector machine in order to see whether we could find a specific descriptor that would make them the difference between the two levels of patient characteristics. So the good thing is that we basically try to optimize a hyperplane that might depend that will depend on the dimensionality of the problem. And here are the results. So first, we had 2 groups of persons. So they all had more or less similar level of osteoarthritis, but some were referred to conservative treatments and others were referred to surgical treatment. So first, we look at the groups. And we've seen that the treatment decision classifier, whether conservative or surgical, largely depending on pain. The accuracy is not super high, but it already says something about the orientation of the data. So knows that we know that treatment decision can be is influenced by pain. We say, okay, so what would influence pain? So then we build a pain classifier, and we see that we actually then have two important factors. So on the one hand, we have catastrophism, and on the other hand, we have effusion. So effusion would be like the swelling of the synovial joint. So somehow it's a consequence of inflammation. So we look at synovitis. So synovitis is the thickening of the synovial membrane that is directly then linked to the dysfunction of the endothelial cell layers and due to inflammation. And we see that synovitis classifier largely depends on effusion with very high accuracy. So now we say, okay, so what can link catastrophism and effusion that are both then involved in the pain classifier? So we took the variable of sensitization. So sensitization is when the patient will start to feel pain more constitutively than normal baseline, no? That in a healthy situation. And we split up the patients in two levels of sensitization, so low sensitization and a high sensitization. And it was very interesting to see that catastrophism was very important in the low sensitization classifier, whereas effusion was the predominant factor in the high sensitization classifier. So there is a switch of key patient characteristics that depend on the level of sensitization. So it seems that when you arrive to sensitization, it's very difficult to then to control the pain of the patient. So it seems that controlling synovitis and effusion seem to be cornerstone to manage the perceived pain and then optimize treatment decision based on proper management of catastrophism, because you don't enter in this area of high sensitization. So you basically focus on catastrophism that might require more psychological treatment management of the patient, and it might be cornerstone actually to lower the rate of surgical treatment of osteoarthritis, which cannot be undone. And as one of my friend, a rheumatologist, says, when you surgically operate a patient with rheuma, actually you're giving the patient a second disorder, which is then the invasive implant, and you need also to manage the invasive implant. So now we wanted to know whether things related also to mechanical loads. So we've seen that the pain classifier further depended on gait velocity, and in statistical analysis, multivariate statistical analysis. So we have also seen that catastrophism seem to be correlated with increased limping gait. So we have here data tells us that something is happening with mechanical loads, but at the clinical data level, we couldn't find any direct link between gait and effusion or synovitis. So all the classifiers that we have tried in that sense were basically not working. So the information is probably too deeply hidden, has too many interactions, and in order then to see things only through statistical correlation analysis or machine learning classifiers. So we decided to enter the matrix. So entering the matrix is to say, okay, so we know that inflammation is important. So know how inflammation relates to mechanical loads. That's an important question here because in terms of patient management, it might allow them to personalize physiotherapy management in case also of anti-inflammatory treatment, for example, through biologics. It might also help to anticipate the real efficiency of biologics, for example, the injection of autologous anti-inflammatory factors, depending on the anticipated mechanical environment in which then these anti-inflammatory factors will be injected. The problem is that when we come to articular cartilage, some biological systems are well characterized, and some others are not. And actually, Ralph already made this observation. And when we arrive to cartilage, relatively few things are well defined and are quantified. So if you look at all the catabolic pathways, you have much more information than if you look at the normal regulation of the cartilage. But if you want to do model, you always need to have a baseline. You actually make comparisons between two types of simulations. So if you don't have the healthy baseline, then you have a problem. And you see that here we have a lot of question marks. The network of interactions is much less populated than here. So we decided to go to high-level modeling. So high-level modeling is basically representation of knowledge through a series of activations and inhibitions. And through that, you build graph-based models. Okay, so our graph-based models was looking to link synovitis or effusion, so inflammation, to mechanical loads. So we seek then this interaction, and we want to further relate this interaction with pain and with the regulation of structural proteins of the cartilage, the collagen type 2 and the proteoglycans. So we selected, so we decided then to focus on the biochemical regulation of the key processes of OA, focusing on soluble proteins. Why soluble proteins? Because these are those that you can measure in the synovial fluid. So we decided to not enter into the intracellular regulation, transcription factors, etc., but to really stay at things that can be relatively easily measured, either in vivo or in vitro. So here you have more or less a list then of the soluble factors that are involved in the maintenance of cartilage, and then the soluble factors that are involved in the destruction of the cartilage. And we targeted these factors, and we did the manual curation of 63 specialized journal articles about articular chondrocyte regulation and osteoarthritis. And here is then the network, the knowledge-driven network that came out of this exercise. So you have in green activating ages, and in red, so inhibiting ages, and all the nodes are soluble protein, except then of course the matrix protein that are most likely related to mRNA expression at the end of the day. So the thing is that when you have such high level network, you can't really define rate constants in order to mathematically then describe the inhibiting and activating relationships. So we took a previous work of Ioannis Xenarios, who will present next, and and they built back in 2006 an extremely useful model, which is a continuous interpolation of Boolean regulatory networks. So you take a graph-based knowledge and you're solving by using Boolean rules, so you don't have rate constants. So your nodes, depending on the rules that you decide to apply, are either 1 or a 0. So if you explore then the whole set of steady states of these Boolean regulations, so you can start then to work out continuous regulations of the nodes. And this is what you have here. So here you have continuous regulation of the nodes depending on the time. And this time is an artificial time if you don't have the rate constants. But since it's actually driven from Boolean networks, you don't really have then the rate constants. What you have is that you have a total activation factor in this equation, and that depends on the topology of the network, depending on activation strengths and concentration of activators, inhibition strengths, and concentration of inhibitors. And you have 3 types of figure. You have an equation if you have a mix of inhibitors and activators for specific nodes. For example, for this node here, you see that you have a mix. And you have one equation if you have only activators and one equation if you have only inhibitors. If you look at the terms, so basically here you have activators, here you have inhibitors, and this is what you find out here. So when you solve then the system for each node, actually the good thing is that you have a continuous node regulation between 0 and 1. So since the time is artificial, you don't really care about the transient evolution of the nodes unless you have specific time-dependent information through which you can inform these equations. Okay, but by default, you basically care about the steady state. The very good thing here is that the steady state actually can evolve over a continuous scale between 0 and 1. And that's great because you can then directly compare these steady state results with measurements of multiplexing proteomics, with measurements of relative mRNA expressions in in vitro setups. So we use then this equation to solve our knowledge derived network. To do that, we first defined a baseline for the network. So for this, we ran 100 simulations with randomly selected initial conditions, and to have a mean behavior of an average behavior of the network. Then we perturb the network with pro-anabolic or pro-catabolic initial conditions. So typically pro-anabolic then would be growth factors and or anti-inflammatory factors, and pro-catabolic would be then pro-inflammatory factors. And this is what we obtain. These are our steady states, and these are the different molecules we're looking at. We see that in this network, we have no difference between the baseline and the inflammatory stimuli, which is not a good news, because it means that by default, our network that aims to represent the behavior of the articular chondrocyte is not able to have a biosynthetic activity. You always have full expression of pro-inflammatory nodes, such as interleukin-1 beta. You don't have expression of structural proteins such as proteoglycans or collagen type 2. So we then compared, we went back to the literature, and we looked at what would be then the expected responses of the chondrocytes, so according then to different stimuli. So thanks to this kind of analysis, so we identified our problematic nodes within this network, and we went through the high throughput tool in order to locally enrich our networks with complementary proteins. And this is the result of this enrichment. So here in bold, so you see all the proteins that have been then added in interactions with the different nodes that we have. So meaning that we can add ages and we can also add proteins. In general, we mostly found anabolic interactions, and we also found specific interactions that were not documented in the literature as specific to articular chondrocyte. So here, the take-home message is that the literature you have actually has a bias, because it mostly focuses on the problem and not on the healthy state. So here we had a bias towards catabolism. And so not everything is documented. So you might need to go through more generic sets of literature through automated methods than in order to enrich the things. So we enriched our network, and then we run our simulations. And no good news. So actually at baseline, so we have our structural proteins that are expressed. We have anti-inflammatory factors that are expressed. We don't have proteases. We don't have pro-inflammatory factors. And when stimulated with inflammatory stimuli, so basically all what should be pro-inflammatory is activated. So once we've done that, we actually then need to validate this thing. Okay. So before validating it, we wanted to calibrate against proteomics data, and it allows to pass from a knowledge-driven network to data-driven network that we can validate independently with other data. So we did our calibrations by using genetic algorithms, and we obtained an optimized network. So again, the optimized network has significant differences between baseline inflammatory stimuli, with difference, curiously, that this optimized network had no activation of anti-inflammatory nodes, specifically IL-4, that is believed to be constantly activated. So this is our pipeline. So we have our first knowledge-driven network based on the literature, the second one based on the enrichment, and then we have the calibration, we have our data-driven network, and then we can validate independently this data-driven network and assess the relative errors that we have. So after the optimization with different experimental sets, with training set and with independent validation set. So overall, we see that the mean response per stimuli of our network is very good. So both in the training, so our genetic algorithm works well, and in the independent validation, meaning that we're limiting basically overfitting effects. So now I'm explaining that, and then I will go then to the conclusions, because we'll be very short in time. So now we want to link that stuff with the mechanics. So we went actually back to the knowledge base, and now we focused on something a bit more complex, which is mechanotransduction. So we made a list of all the mechanotransduction mediators that chondrocyte might have. And here we actually then mapped all the transcription factors, so basically the most relevant proteins that we found in the literature associated with the cell regulation pathways, so down to the expression of specific genes. And once we had then this expression of the specific genes, so we actually then used the activation of these expressions to activate the nodes of our data-driven biochemical network. And again, we tested that. So now we have actually a system of networks. We have two coupled networks that work together, and we define a new baseline. We define so the new baseline So included physio-osmotic pressure, so the basic osmotic pressure that you have within the articular cartilage. And we also induce so pro-inflammatory stimulation that would typically be due to, for example, synovitis. And we also then introduced mechanical stimulation related with static compression of which shear stress that we know chondrocytes don't like too much. So what was extremely interesting here is that, so first of all, in terms of transcription factors, so in the baseline, so the cartilage related transcription factors were present, ID2, FOXO SOX9, which reflects then normal activity. And also, actually, the presence of the physio-osmotic pressure and the coupling it has with biochemical regulation networks then activated actually our anti-inflammatory cytokines and in a constitutive way. That was, and it was very good news because actually this activation of IL-4 has been related to specific membrane receptors that are calcium sensitive and are so depend on the osmotic pressure. Of course, we also were able to predict biosynthetic activity. When we had inflammation, so we lose then the biosynthetic activity, we activate so our pro-inflammatory factors, we activate our proteases, and we start to also have signs of possible hypertrophy with bone morphogenic proteins. And when we have undue mechanical properties, so we have more or less the same, but with a milder effect. But we basically switch off then the biosynthetic activities in terms of structural proteins. Okay. So now if we want to upscale things, and this is where combining everything becomes extremely interesting. So we have our gate analysis through which we determine actually reaction forces at the knee joint. We have then medical images, MRI, through which we can personalize by a finite element model of the knee joints with all the represented tissues, by mesh morphing, by scaling. And we can then make our simulations, and we have specific constitutive equations for the different tissues that include then the flow of waters, the volume content of waters, and also the composition in terms of protoglycans and cartilage. Meaning that after the simulation, so here you can see the meniscus that are moving, you can see the knee joint that is being squeezed by the load. We can achieve then the breakdown of stress that is due to osmotic pressure, that is due to solid stress that will be mostly pure compression or traction, and then the stresses that is within the pressurized fluid within the tissue that will be hydrostatic. And now we know then through this, thanks to this breakdown, we know so which actually mediators of mechanotransduction we can activate so in this network model, and we can then finally close the loop. So I had the stuff about the intervertebral disc. I have decided to present the most recent things, and where maybe we don't have the best achievements or the most accomplished work. We have that really in the spine system and intervertebral disc modeling. But I thought that it was really interesting and to see how network modeling then could be combined in a relatively understandable way based on knowledge representation, and to show how this could be then related with more physics-based modeling in order then to have a representation of all then these different interactions that are within the mapping of biochemical and mechanical interactions that I've shown at the beginning of in the first slides. Here, basically, you can see then the type of equation that we have to make the tissue dependent on protoglycan contents, on collagen, water contents, etc. And this is typically what is implemented here. For the interpretable disk, basically have a full pipeline. We have also the agent-based scale, which allows them to compatibilize the scales between the finite element model, which is typically at the millimeter scale, and then the cell scale, which is submicrometric. So here we have collections of cells that can be represented within the volume. So now I will skip the detail of that. If within the questions you want to have more detail, and I'm happy to come back to these slides about spine and intervertebral disc. Just wanted to mention that now we have a large European project, which is an innovative training network, European training network. That is, and actually this summer school is part of this project, where we are really trying then to push things further with the collaboration of 19 international partners. So here we integrate population data with image analysis, correlation of image analysis with multiomics, with bioinformatics tools in order then to relate then the genetic variants to the regulation of the proteins I was talking about. All of that then backed up with cells and organ cultures under different conditions to calibrate and then validate the models. And all that stuff, of course, then will feed image-based patient-specific models and multiscale models, such as the ones I have presented. And then by the end of the day, by using high-performance computing infrastructures, then we will be able to simulate different cases informed by real-world data, and we will have a mix of real-world data and synthetic data. So typically, it's synthetic data that cannot be measured in the patient, and have an information about the mechanisms I was explaining before. And based on that, so we hope to achieve actually higher level of interpretable patient stratification. So based on this data, on this real world data, and then on the interpretable data that will come then from the multiscale simulations, partly informed from real world data and partly informed from mechanistic controlled experimental data. And I think that's it. So I would like them to finish then to thank actually then all the people of the teams that are always behind them this work. You see that they are distributing among the different scales and the different modeling technologies, which is actually a great thing. So yeah, I'm happy to take questions now.
Thank you, Jérôme. Are there any questions? If you'd like to pop them into the chat or raise your hand. But maybe if I can start whilst everybody's having a think. I was interested in the population data and your sort of classifiers. And I was just wondering whether you've considered, because fusion was clearly something that discriminated in your OA phenotype, whether you could use your classifier to discriminate against those OA patients who have quite a high inflammatory environment versus a low inflammatory environment. You know, thinking about, you know, a lot of people now talking about a sort of a subtype of OA being an inflammatory OA phenotype. And I was wondering whether your classifier would allow you to discriminate in your patient population.
Yeah, so this is the ID, and this is actually the PhD thesis of Maria, who chaired your talk. So Maya is developing then all these network models that I have shown, that I have presented during her PhD thesis, and though she will soon then connect these models with synovial fluid proteomics measurements that we have for all the patients of the cohort. So it's actually great data, and we hope that with this we will be able to enrich a little bit the classifiers and to find better things. So we can either enrich a classifier so with the raw data of the proteomics measurements, or we can enrich the classifier with a combination of these raw data plus some synthetic data that we have used to interpret this raw data. So that's something that will come then in next step. So the first step is actually to duly validate then these models that are presented. So right now we have verified that things are working in the correct direction. And but we are still in a process of refining.
Because I can see that being extremely powerful if you think about biologics, and some patients obviously do respond very favorably and others don't, and whether you could use that then as a predictor for sort of personalized medicines or treatment strategies. That'd be really powerful.
Yeah. Actually, we've made a step in that direction. So we gathered from the literature information from a clinical trial with autologous injection of autologous anti-inflammatory cytokines. So they were IL-4, IL-10, IL-13, IL-1 receptor antagonist, and a cocktail of this kind of proteins. I'm not sure I have all of them in memory. And we found something very interesting. We found that under certain circumstances, this injection was able to particularly inhibit the prostaglandin 2 and vascular endothelial growth factor nodes that are related to nociceptive signaling, so to pain, inflammatory pain. However, the restoration of the structural proteins, of the expression of structural proteins, was extremely difficult to achieve, at least with our network. And in order to properly restore this, you really need to eliminate all side inflammation, which I think is extremely difficult to do, because actually you have then the synovial membrane with the endothelial cells that are responding to the systemic system that is also responding to what's happening in the joint. So if you only inject the anti-inflammatory factors in the synovial joint, actually you're not completely shutting down the inflammation. So, and it might explain the variability in some patients. And globally, what they found in this clinical trial is that a patient had pain improvement, but no radiological improvement. So basically, then the model was going also in that direction. But if you play with the initial conditions, you get actually then different results, which is interesting because you can interpret this. And now we need to see what will happen with the mechanical loads. So if we have physiosmotic conditions or not, because we're in a degraded cartilage, and we might have reinforcement, constitutive reinforcement of IL-4 and of others. So I think this is the beauty of the model, is that you can try and interpret all those things to have better explanations of what happens in this kind of trials.
And going back to the study then with the OA patients, do you remove any comorbidities? Are the patients purely suffering, do you know what I mean? Suffering with OA, and there are no confounding factors?
Yeah, I mean, that's tricky. That's tricky because actually one of the conclusions of the medical doctors of the project is that after 3 years of project is that there might not be any idiopathic osteoarthritis. So I think at the beginning, they were extremely careful, and the patients have been selected very carefully to avoid, to eliminate any obvious signs of secondary osteoarthritis, so due to previous injury. Of course, people who had chronic disorders or diagnosed or declared chronic disorders were not selected either. So we try then to ensure that we had the cleanest cohort as possible towards what we believe would be idiopathic osteoarthritis. But yeah, that's so people have a certain age. I mean, I think the mean age of the patients was nearly 70 years old. So you can't eliminate everything, otherwise you don't have patients anymore. And actually recruitment was a real struggle because we were extremely selective.
And what would you expect to see if you had a PTOA population?
Pardon?
What would you expect to see if you had your patient population that was purely, sort of secondary OA in, a post-traumatic OA phenotype?
Well, I think post-traumatic osteoarthritis is relatively easier to interpret because you have an initial shot of TNF alpha, and TNF alpha is a real powerful trigger of inflammation. So if you combine then this initial shot of TNF alpha with activation of macrophage 1, and because of age, you always have low-grade inflammation. And then this low-grade inflammation may impede actually the taking over of macrophages type 2. I think things are straightforward. I mean, you have all the numbers to enter in an endless catabolic loop. Of course, this is my own interpretation of the things. No, I think secondary osteoarthritis is easier to interpret. Of course, it's not easier to manage. It's as tricky as the first one, as idiopathic to manage, but at least easier to interpret. So right now we are at that level because since we don't have real biologic treatments or tissue engineering treatments, so we're not at that level of questioning, what who would be the target.
It's fascinating. I don't know, are there any other questions? I'm conscious of time as well. I'm not quite sure where we are with time. I can't see any hands raised. I mean, I could ask questions all day, but I'm not sure. Should we have a 5-minute quick break? Is that sensible before the next session?
Yeah. I think we should. I think we should have, and of course,